So here, when our skin gets damaged, which is part of our first line of defense, our second line of defense is going to become activated. Here, the mast cells that are right below the surface of our skin become damaged. Now, these damaged mast cells are going to release a chemical called histamine. Now, histamine is going to cause a response in the blood vessels nearby, the capillaries. So the capillaries, in response to histamine, are going to dilate. So the capillaries are going to expand and dilate, which causes the walls of the capillaries and the cells that make them up to kind of spread apart and become more permeable. So they become kind of like leaky. And therefore, fluid from inside of our blood is going to leak inside the tissues surrounding the capillaries, causing swelling. Now, because there's more blood to the area and there's fluid leaking from our capillaries, we get signs of redness and swelling. And there's also some little chemicals called pyrogens that will activate change in temperature, so an increase in temperature or resulting in heat. These three things are all signs of a local inflammation. Now keep in mind, also, the walls of the capillaries, once they're dilated, they're going to release a chemical called chemokines. Now, the reason for these chemokines, if you think about it, right here, you have a break in the skin and the cells are damaged. Now, most likely, when your skin is broken, this bacteria or pathogens that were on your skin or on the thing that poked you or broke your skin allowed bacteria now to enter into the tissues. So it makes sense that you need to bring white blood cells to that area. So when we look at this big picture, the broken skin damaged the mast cells. The mast cells released histamine. Histamine caused the capillaries to dilate. The walls of the capillaries that dilated released chemokines. Chemokines attract white blood cells, phagocytic cells, that are going to engulf the bacteria. Because most likely, if the skin was broken and the cells were damaged, there's going to be an entrance of bacteria or other microbes. So that would make sense to bring phagocytes to that area to engulf and clean up the tissues. So here you have these white blood cells. These most likely are neutrophils, which are about 70% of your immune system's white blood cells, and they're going to start to engulf and clean up the bacteria. You also have blood clotting factors that are going to travel to the area to form a scab. So now these blood clotting factors serve two purposes. One would be to protect it from new bacteria entering, but also preventing further bleeding, so you actually stop the flow of blood. Now the bacteria will be destroyed by the by the neutrophils, white blood cells. The area will get cleaned up, the tissues, the swelling will go down, the blood vessels will return back to normal, and now your local inflammation is gone. So this right here that we just saw is the second line of defense, called the inflammatory response. It is nonspecific. However, it happens if the first line of defense has been passed, I guess you could say. So when we look at how phagocytosis works, though, so if here's a phagocyte engulfing a bacteria, it brings it in by the process of endocytosis. So here we have a vesicle surrounding that bacteria. Now we have our organelles called lysosomes. Now, if you remember from our cells chapter, lysosomes are like the cleanup crew of the cell, the garbage truck. They have hydrolytic enzymes inside, which are going to break apart macromolecules or carbohydrates, lipids, proteins, etc. So here the lysosomes are actually going to fuse with the walls of that vessel that had the bacterium. So if you think about like when you blow balloons, how two balloons might, not balloons, bubbles, how like two bubbles mid-air might bump into each other and they fuse and become one. That's kind of what's happening with the membrane of the vesicle. It kind of fuses with the membrane of the lysosome since they're both made of a lipid bilayer. So then the lysosomes are going to release their different enzymes into that vacuole there. And then the bacterium or the microbe is going to be destroyed and then sent out of the cell by exocytosis. So the two ways that microbes are destroyed in this process is by nitric oxide, which can poison the microbes, and there's also lysozyme, which is that enzyme that's going to degrade the cell walls of the bacteria. And then there's other enzymes inside of lysosomes that play a role in degrading molecules. So that's phagocytosis.